Our next speaker is Dr. Marian Schmicke from the Martin Luther University. She's going to be talking about the interaction between the somatotropic axis and local IGF system and the role of the IGF-1 binding proteins.
Okay, thank you very much for your kind introduction. So my question first is, can you hear me and can you see my presentation?
Yes, we can hear you and we can see you.
Okay, wonderful. So thank you very much for organizing this event and for giving me the opportunity to give a talk here. So unfortunately, as already mentioned, we in Europe were not able to come. So we are very sad not to be here at the Atlantic Ocean, but sitting right here in Germany. So it's the eastern part of Germany in Halle. This is a picture of the city where we are now. Halle is a very old city, first mentioned in the year 806, and it's an old salt town. Today, it's the largest city in the regional state of Saxony-Anhalt. And in July 1694, the University of Halle opened. And I'm right here at the moment. So only to give you an impression, this is one part of the university called Weinberg Campus. And I'm sitting right here in the red building over here. Here's the office, and I would like to give you the talk today about the somatotropic axis. We already heard a little bit about this, and especially about the potential role of IGF-binding proteins while combining the endocrine part of the somatotropic axis with the local IGF system. So therefore, I would like to introduce both systems, very simple, and then go over to the binding proteins in physiology and pathology. In the second part of my talk, I would like to depict a model of interaction, and I would like to do it within the transition period in dairy cows and early pregnancy, so early lactation and early pregnancy in dairy cows. And I would like to show you some data we have obtained within this period with regard to the binding proteins. So as most of you will know is that growth hormone is secreted by the hypothalamus, primarily controlled by two hypothalamic peptides, growth hormone releasing hormone and somatostatin. And this controlling leads to a pulsatile pattern of growth hormone release. Growth hormone itself is produced and secreted by specialized cells in the anterior lobe of the pituitary gland. And after binding to a receptor, to the growth hormone receptor, IGF-1 is produced. There are many organs expressing the growth hormone receptor. The liver is one of the organs which is expressing the receptor in a very high density. And when IGF is produced, we can find and measure IGF-1 in the circulation, and it has a classic negative feedback loop on the hypothalamus. We should not forget ghrelin. Ghrelin is produced in the abomasum and duodenum in dairy cows, or in cows and ruminants, and has a positive feedback on growth hormone release by the pituitary. On the other hand, we have not only the liver producing IGF-1, but many other tissues. So for instance, the adipose tissue itself is able to produce IGF-1, the muscle tissue itself, and also the bone. So when we measure IGF in the circulation, so total IGF, it's about 75% liver derived, and the rest is coming from other tissues. The production of IGF-1 from other tissues can be growth hormone dependent and independent. So tissues which are producing IGF-1 partially growth hormone dependent, this is the adipose tissue, the muscle tissue, the bone, the liver, and the kidney. On the other hand, we have growth hormone independent IGF-1 production, for instance, by the brain, the heart, the gonads, and partially also the placenta is producing itself IGF-1 growth hormone independent. And IGF-1 was functioning, working in the combination between primarily local effects, so paracrine or autocrine, and also do have some endocrine effects as the negative feedback loop to growth hormone release. So therefore, we always have to deal, when talking about the IGF system, about the endocrine part of IGF and the local part. So on the right, on the left-hand side here, the somatropic axis, and on the other hand, we have the local IGF system with a target cell, which might be the liver, a hepatocyte, but many other cells are, as already mentioned, expressing the growth hormone receptor. For instance, even immune cells do express the growth hormone receptor. On the other hand, on the target cell, we can also find the respective receptors for IGF-1, of course, also IGF-2, and there's also a hybrid receptor which can bind IGF-1 and IGF-2. And in this concept, we should not forget insulin. We heard about insulin already, and insulin binds to the insulin receptor, but can also bind to lower affinity to the IGF receptors, and vice versa. IGF-1 is also able to bind with lower affinity to insulin, to the insulin receptor. And we have locally produced IGF-1 and IGF-2, and we should not forget the transport of these hormones. And not only IGF, but many other hormones are transported in the blood by specialized binding proteins. And not only local IGF binding proteins binds IGF-1, but also in the circulation. So when we talk about a connection, a potential connection between the endocrine side of the somatotropic axis and the local tissue level, we should talk about the IGF binding proteins. These binding proteins, we know 7 binding proteins, they all belong to a protein family, IGF binding protein 1, 2, 3, 4, 5, 6, and 7, but there are also reports on binding proteins which are not expressed in such high concentrations. So there are also reports on IGF binding protein 8, 9, 10, 11. All these binding proteins are structurally very similar, and they all bind IGF-1 and IGF-2, with one exception. IGF binding protein 6 has a distinct preference for IGF-2. And there is also a special protein, which is called Acidlabel subunit, which forms a complex to one of these binding proteins, to IGF binding protein 3. And what is important is that those binding proteins modulate the IGF action by functioning as IGF carriers within the circulation, and with that modulate important biological processes. So including cell proliferation, survival, migration, senescence, and also angiogenesis. There are also reports available that shows that those binding proteins do not exclusively inhibit IGF action by binding of IGF-1 or 2, but there are also reports that shows that those binding proteins are also able to potentiate IGF action. So when doing an alignment between the different species, cow, pig, together with human protein sequence, you can see that we have a high homology between those binding proteins, between species, so they are highly conserved, especially IGF binding protein 4 with 97 or 96% homology between cow, pig, and human. And this is also showing the importance of those binding proteins within the endocrine system. 90% of IGF in circulation is bound to a complex of, I already mentioned it, the Acidlabel subunit, and IGF-bp3. A smaller proportion is also bound to another complex, between the Acidlabel subunit and IGF-bp5. And what is important to know is that those ternary complexes are unable to cross the vascular epithelial layer, so they are forming an IGF reservoir. So when we usually use immunoassays to detect the total IGF-1 concentration, we usually do ethanol extraction and cleaving all the IGF free from the binding proteins so that we can measure total IGF irrespective to which binding protein IGF is bound. On the other hand, we also can find other binding proteins than 1, 2, 4, and 6 also present in blood. And when IGF-1 is bound to the smaller binding proteins and not to the complexes, these smaller IGF-bp complexes can cross the vascular epithelial layer and with that may deliver IGF-1 to the target tissue. So this Acidlabel subunit, therefore, is an important protein complexing IGF in the circulation and also inhibit the transport to the local tissue site. This Acidlabel subunit belongs to the lysine-rich repeats family of proteins and increases the half-life of IGF in circulation. So normally IGF-1 will have a half-life of 12 minutes, but when it's bound to the complex with Acidlabel subunit, the half-life is prolonged to more than 12 hours. And only free IGF is biologically active, and therefore it makes sense in the circulation to bind IGF-1 because, as I've mentioned, it can also bind to the insulin receptor and can lead to hypoglycemia in high concentrations. So therefore, the binding of IGF to the binding proteins is a protection against insulin-like effects. Binding proteins and also the Acidlabel subunit is mainly produced in the liver. There are some important or interesting reports in the literature, but mainly only from rat hepatocytes, that one of these proteins forming the complex, the Acidlabel subunit, is localized exclusively in the hepatocytes, whereas IGF-bp3, however, is expressed in the non-parenchymal cells, endothelial and Kupffer cells. Also, IGF-bp2 is expressed in high concentrations in the liver and hepatocytes. So this is what we also could show, that IGF-bp2 is produced in bovine hepatocytes, and the expression is suppressed by insulin in vitro. So we have on the one hand the binding proteins which are produced in the liver and we can find in the circulation, but on the other hand we again have not only local expressed IGF-1, but we also have local expressed binding proteins, and they also able to modulate the IGF action then on the tissue level. Those local IGF production is important because these local binding proteins also hinder the interaction of IGF to the IGF receptor and therefore modulating IGF function at the tissue level. We know already two mechanisms of IGF release at the tissue level, which is on the one hand extracellular matrix binding. So for instance, IGF-bp5 is normally incorporated intact into the extracellular matrix. And if it's released from that, it can be modulated the IGF function at the local tissue level. And the other mechanism we know is IGF-bp proteolysis. And because we have done some research in farm animals on IGF-bp proteolysis, I would go a little bit in detail with that. IGF-BPs are susceptible to proteolysis by a range of proteases, and those proteases, which are normally more expressed on the tissue level, cleave the binding protein, releases IGF-3 so that this can bind to the local tissue level. This is a typical structure of those binding proteins. These binding proteins do have an N-terminal and C-terminal region, and near the linker region in the middle of the protein, and nearby the linker region, we can find a proteolytic cleavage region where this binding protein can be cleaved by proteases. This IGF-BP proteolysis is firstly discovered in pregnant women because IGF-BP fragments were found in blood from pregnant women, and a pregnancy-associated plasma protein A was the protein which was the underlying cause for this IGF-BP proteolysis in women. This PAP-A is cleaving all binding proteins, but a high proportion of IGF-BP4. IGF-BP4 is also known as an inhibitory binding protein. The cleavage of IGF-BP4 is dependent on IGF-1, and it makes maybe sense because during pregnancy we need IGF for at the local tissue, maybe for growth, for fetal growth, for embryonic development next to IGF-2. So therefore it might be logic that an inhibitory binding protein is cleaved. And there are also other proteases, including pepsin, catepsin, or other matrix metalloproteinases, which are able to cleave the binding proteins and release IGF-3. So we've heard about binding proteins, the complex between binding proteins within the circulation and on the tissue level, and therefore the indirect actions on IGF. So they are important for bioavailability in order to make an IGF reservoir in blood, and they are also functioning as carrier proteins. But what is also known from recent literature is that those IGF-binding proteins do also display direct effects. So these direct effects are on the one hand possible by IGF-BP receptors on the target cell. They also are able to have an impact on the calcium influx in the cell. And on the other hand, they are already also, or it's also possible that those binding proteins enter the cells, and transcriptional effects following a nuclear entry of binding proteins is already described in literature. So therefore, we do not have indirect actions on IGF-1, but also direct effects of the binding proteins itself. And I've summarized some examples of these direct effects which binding proteins could have. These reports are mainly derived from human medicine, or but not from farm animals until now. So for instance, IGF-BP1 and 2 direct effects of IGF-BP1 and 2 are directly linked with insulin sensitivity. IGF-BP1 has an impact on placental function and fetal growth directly, and IGF-BP3 interacted with receptors, including PPR gamma or the vitamin D receptor, which affects apoptosis, proliferation, and also differentiation. IGF-BP1 and 6 were able to be increased by pro-inflammatory cytokines, and this might divert energy towards the immune system, parallel to suppressing IGF-mediated growth. IGF-BP4, again, interesting because it inhibits angiogenesis, and we of course know during pregnancy and placenta forming, it's very important. Angiogenesis is very important, so maybe this is the next point why it's maybe important that this binding protein is cleaved during pregnancy. And IGF-bp6 promotes monocyte and lymphocyte chemotaxis and may also play a role in the adaptive immune response. So therefore, to summarize, IGF is derived from the liver and other tissues and complexed in the blood to IGF-bp3 and the Acidlabel subunit, or IGF-bp5 and the Acidlabel subunit. IGF-bp regulates bioavailability of IGF, and not only in the blood, but also important to regulate the function at the tissue level. And IGF-bps do have also direct effects. So this was a short introduction into the field of binding proteins. And I would like now to come over to the results from farm animals. I have focused in my talk really only on binding protein measurement in blood, because when talking about the link between the somatotropic axis and the local IGF system, IGF-bps in blood play an important role. Of course, there are many other reports available where IGF-bp expression is already detected in specific cells. And I would like to give some results about IGF-binding proteins, as I already mentioned, during, on the one hand, transition period from late pregnancy to early lactation, and on the other hand, during early lactation, so as possible, or with a possible impact on embryo-maternal communication and the potential function of IGF-bps during pregnancy. So starting with the somatotropic axis, you most of all will know about the uncoupling of the somatotropic axis that was already mentioned in the first talk. So what we know is that hepatic growth hormone expression and growth hormone binding declined, IGF production decreased, and therefore the negative feedback to growth hormone is diminished, and therefore in the late pregnancy or early lactation, growth hormone secretion is increased. And we already heard in the first talk for what it's important as direct effect on tissue level, lipolysis, glucose transport, insulin resistance, as important effects at the tissue level. So what we have measured within this uncoupling of the somatotropic axis was not only the decrease in IGF-1, in total IGF-1, and the increase in growth hormone, what we already saw in the first talk, but we also measured the binding proteins. So as you can see here on the left-hand side, the concentration of IGF-bp3 before parturition, and on the other hand, the concentration of IGF-bp2 before parturition. And as you can see, the concentration of the binding protein 3, so this what is forming the complex and complexes IGF in the circulation, this binding protein concentration decreases, whereas on the other hand, the IGF binding protein 2, which is more a transport binding protein, increases in late pregnancy and maybe deliver IGF to the target tissues. So therefore, it's maybe important for late fetal growth. We ask ourselves whether there is maybe this proteolysis in blood of pregnant cows here in late pregnancy, but we could not confirm any proteolysis here in late pregnancy. But what we have detected is a less hepatic mRNA expression of those complexing binding proteins, IGF-bp3 and Acidlabel subunit, as you can see here, before parturition and after parturition, the content or the expression, the hepatic expression of IGF-bp3 and also of the Acidlabel subunit decreased. Whereas on the other hand, similar to the blood concentration, the IGF-bp2 production in the liver increased while comparing liver biopsies here in this case before and after parturition. So we see an increased hepatic mRNA expression of IGF-bp2. Therefore, we can summarize during the transition period, there is not only this classical change in IGF-1 and growth hormone, but moreover we can also see that the ratio between reservoir binding proteins, BP3 and Acidlabel subunit, changed towards the transporter binding proteins. Yeah, there is a switch which may deliver those IGF also to tissues, to local tissues, which might be important for fetal growth. Interestingly is that we know from literature that normally low substrate availability decreases IGF-3 and 2 in the intermediate term to limit IGF bioactivity. However, this is not the case in late pregnancy, and this might be a potential effect of estradiol on IGF-BP2 production, which is known that this increases IGF-BP2, at least in liver. Coming to early lactation, to embryo-maternal communication, I'd like to show you some recent data we have obtained. So the hypothesis is from some researchers that interferon tau, we heard already about this hormone, which is a pregnancy signal, the classical pregnancy signal in cattle, not only acting local, but maybe also have an endocrine effect. So the current hypothesis is that maybe exocytosis or vesicles are able to transport it to the liver. And we have already detected the classical interferon tau gene expression like MX1, 2, ESG15, and ORS in liver biopsies obtained from pregnant dairy cows on day 18 compared to non-pregnant. So therefore, there is a potential effect of interferon tau on hepatocytes on the liver. And we ask ourselves whether this signal may modulate maternal IGF-binding protein concentrations. So for that, and of course further studies, we set up hepatocyte cell culture, so we can gather hepatocytes from adult liver and cultivate those hepatocytes in between two layers of collagen in a monoculture. And for that experiment, we also cultured Kupffer cells in direct or indirect contact to the hepatocytes. So as you can see here, those bovine adult hepatocytes in the culture are very nice and stable for at least 7 days. And we can measure or we can do some experiments on that here. This is a two-step collagenase perfusion where we can obtain or gather them from the hepatic part, from the lobus caudatus in this experiment. So I would like to summarize some of the results we have obtained here. So what we can see is that in contrast to the control, interferon tau in a concentration of 0.6 nanograms per milliliter is interestingly suppressing IGF-BP2 production in hepatocytes in the monoculture, but this is the effect is diminished in the co-culture. So therefore, there's really a potential effect of the embryo signal on the IGF-BP system in the maternal blood. And thirdly, I would like to go in detail with the IGF-binding proteins throughout pregnancy. So what I've mentioned already is that we saw from human pregnancy this very strong proteolysis of binding proteins. And we ask ourselves whether there is also this binding protein proteolysis in cows. So for that, we did the Western ligand blot. So with that, we did the normal Western blot and afterwards incubated it with radio-labeled or biotin-labeled IGF. So therefore, we can measure binding of marked IGF to the respective binding proteins here. And I'd like to go in detail with that. So I'd like to to start with the human serum, so that was our control. We spiked this serum with recombinant bovine, recombinant human IGF-bp4. And what you can see here is that this is a sample before incubation, 24 hours at 37 degrees. So we can detect this binding protein before, and after incubation, this is not detectable anymore. And this is due to this high concentration of proteases in human blood during pregnancy. So what you also can see is that we do not have any binding of IGFs to any binding proteins here in this Western ligand blood in human pregnancy. So therefore, they are completely proteolized. On the other hand, as you can also see when we do this experiment with a cattle serum from the first, second, and third trimester, you can see here the recombinant IGF-bp4 before and after incubation, and it's lower after incubation, but it's still there. And also the binding to binding protein 3 and 2 is detectable in cattle pregnancy, what is not the case in humans. So therefore, we concluded from this study that we do have proteolysis, but it's not comparable high to human proteolysis. And this is also underpinned, this result, by detecting those binding proteins in blood. As you can see here, the concentration of IGF-binding protein 2 during early pregnancy, so the first 100 days of pregnancy in dairy cattle, and the IGF-bp2 concentration increases during these first 100 days of pregnancy. In contrast, IGF-bp4, this concentration stayed more or less comparable here, and we do not detect any sharp decrease as we have expected maybe from human medicine. On the other hand, so that was a study where we have compared not pregnant cows, but also cows which have late fetal loss. And as you can see here, the IGF-bp concentration in those cows with fetal loss are distinctly higher, IGF-bp2 and 4 compared to the pregnant cows. There's also a nice paper from Claire Waits showing that IGF-bp4 is also higher expressed in the endometrium in cows with severe negative energy balance. However, in this study, all the cows were, well, these are only healthy cows and don't have any severe negative energy balance while starting this experiment because they have a prolonged waiting period in this study. So therefore, I would exclude that those cows here do have a severe negative energy balance. So there must be something else which is the underlying cause, therefore that the binding protein concentration is higher in those cows displaying fetal mortality. And this is not new because there are also reports from human medicine that higher concentrations of or altered levels of the insulin like growth factor binding protein proteinases, but also altered levels of IGF-bp4 are associated to preeclampsia, to pathologic pregnancies, and also to intrauterine growth restriction. So therefore, it may be very interesting for further studies here. And therefore, we also ask whether those binding proteins may also have direct effects on embryo development. So therefore, we incubated cumulus oocyte complexes in vitro during in vitro maturation with recombinant bovine IGF-bp4 in different concentrations, then did an in vitro fertilization and in vitro culture. And what we have seen or what we could show is that compared to the control, there is a lower rate of hatching in those embryos which are incubated with IGF-bp4. So therefore, we can hypothesize that maybe the binding proteins do really have a direct effect on embryo development or embryonic competence, developmental competence. So what we do not know until now is whether the blood concentration of IGF-bp4 or IGF-bp2 is also mirrored by the follicular fluid. So this is an ongoing project currently. So to summarize, we know that pregnancy status affect the IGF-bp concentration. So for instance, IGF-bp2 increases during the first 100 days of pregnancy, but we do not see any severe proteolysis compared to human serum. The higher concentrations of IGF-bp2 and 4 were associated with embryonic fetal mortality in cattle, and we also saw an effect of IGF-bp4 in vitro on hatching of bovine embryos. So therefore, what are maybe the future directions within this field? And one interesting direction I would like to summarize here at the end. We know from a nice study from Lucy that we have functional differences between cattle and pigs within the somatropic axis. So lactating cattle do have the high growth hormone compared with low IGF levels, where those normally have in lactation high IGF and high growth hormone levels. And as we have this in vitro culture of hepatocytes, we could compare cattle and pig hepatocytes and could see that there are distinct differences, at least in the BP2 regulation between these both farm species. So therefore, it's very interesting what is the deeper reason for this biological response in the somatropic axis, and especially within the binding proteins between these two species. So therefore, I would like to summarize overall that IGF-BPs are important regulators of the IGF function, and that IGF-BPs in blood may be the link between the endocrine part, the somatropic axis, and also the local IGF system. Due to proteolysis, IGF-BP or the modulation of IGF-BP production, IGF can be delivered to local tissues. And we may have really in this binding proteins also functional differences between farm animals. And what you can see here, I think also in other hormone systems, the binding proteins are in general underestimated at the moment, also in science. So when you look for IGF-1 and cow, you can see constant publication records here. And if you would check for insulin, there's much more data available on insulin and cow. But if you can, if you check for IGF-BPs, there are some reports, but decreasing reports until yet. And this is not only in our field of research the case in farm animals, but I found a review from human medicine, and also in human medicine, the publication records decreases about binding proteins, irrespective of a very important function of these binding proteins, even in cancer research. So therefore, I think we should focus not only on the hormone, but also on the respective binding protein transporting the hormone to the receptor. And with that, I would like to thank you for your attention, and of course my recent doctoral students, Sandra Andres, Vera Stinsmeier, and Adriana Camacho, and of course several cooperation partners, but especially for the IGF binding protein analysis, I would like to thank Christine and Andreas Höflich, because with both of them I did all the binding protein measurements, and of course thanks for funding. And I would like to be, well, I would be happy to answer questions. Thank you.